This section is about how things get into and out of cells. Obviously, we want nutrients and oxygen and water to go into cells and waste products to come out of the cells so that our circulatory systems can get rid of them. Cells exist in liquids, and one of the most important things that a cell membrane does is maintain homeostasis. There we go, homeostasis again. And it does this by regulating the movement of molecules across the cell membrane. As we discussed today, diffusion is movement of anything from an area of high concentration to an area of lower concentration. A lot of particles and substances that move across the cell membrane are because of diffusion, because we're moving from an area of high concentration to an area of low concentration. Here's a visual example. So in this case, the substance is the little blue dots. It's present in unequal concentrations on either side. So there's a higher concentration outside the cell membrane than inside. The overall movement of the particles then will move into the cell membrane. Particles will tend to move toward the area where it's less concentrated until, and this is the important part, until it's evenly distributed. There'll still be particles that move in the opposite direction, like you see here, but overall the most movement is from high to low concentration. When equilibrium is reached, that means the concentration on both sides of the cell membrane is the same. You can hear the word equal in there, equilibrium. So movement doesn't stop. Particle movement across the cell membrane doesn't stop at equilibrium. It's just moving in both directions in the same amount, as you can see here. So then there's no net change in concentration on either side. Diffusion happens because of random particle movements. There's no energy required. Because of that, it's called passive transport. If something's passive, it's not requiring any energy. Sometimes there are proteins. Remember the fluid mosaic model where I said that there were embedded proteins? These proteins act as carriers or channels to help certain molecules across. This is called facilitated diffusion. And if you look at this word, facilitated, the beginning of the word, if you know Spanish or if you know French, facile means easy. It makes it easy for certain molecules to cross. There are hundreds of different proteins that allow substances to cross the cell membrane. And this also does not require any energy. The inside of the phospholipid bilayer is hydrophobic. We talked about this, water hating. Because of this, water molecules have a tough time passing through. So there are water channel proteins called aquaporins that allow water to pass right through. The movement of water through membranes is called osmosis. Osmosis is diffusion of water. And more specifically, it's diffusion of water through a selectively permeable membrane. And again, because it's diffusion, it's going from high concentration to low concentration. In this experiment, we have a barrier here in the middle, that little dotted line. It's permeable to water. You can see it zoomed in here. So water can go through, but the sugar cannot. So the sugar's too big to go across, but the water can. We start out with a lower concentration on the left-hand side and a higher concentration on the right-hand side. The sugar can't move through, but the water can. So what happens is the water moves to balance out the concentrations to where we end up with equal concentrations of sugar and water molecules. That means that the water has moved across this way, and that causes the water level to rise. So let's go through that again. More sugar molecules on the right-hand side of the barrier. So the concentration of water is lower on the right because there's more sugar there. Water will move across the barrier until equilibrium is reached. And water and sugar are the same on both sides. So water will move to the right, and that will cause the water level to rise. The concentration, if it's the same on both sides of the membrane, is called isotonic. It means the same strength. The more concentrated sugar solution was hypertonic. The diluted sugar solution was hypotonic. So you see those words here. Hypotonic is the diluted sugar solution. Hypertonic was the concentrated sugar solution. Make sure you know the difference between those, and I'll give you some tips how to remember that in class. In order for organisms to survive, they have to balance water, keep that homeostasis going. The net movement of water in and out of a cell creates a force. So osmosis creates a force called osmotic pressure. Because the cell is filled with stuff, I'm going to call salts, sugars, proteins, other molecules stuff. It's almost always hypertonic. It has more stuff in it than fresh water does. So because of that, water tends to move quickly into the cell, causing it to swell. And then the cell might burst. So that's what you see over here. Now the difference is between an animal cell and a plant cell is the plant cell has a cell wall, so that keeps the plant cell from bursting. Animal cell can swell to bursting. So in plants, that central vacuole fills with water and pushes the cell contents out against the cell wall. And that's part of the process that keeps plants upright, defying gravity. In large organisms, the cells are bathed in fluids such as blood that are isotonic, and they neither gain nor lose water. Finally, in a hypertonic solution, water rushes out of the cell, causing animal cells to shrink and plant cell vacuoles to collapse. That's what you see here. Give you a couple of vocabulary terms, one you know and one you don't. In animal cells, when that happens, It's called crenation. And you learned this term already. Plasmolysis in plants. Cells that come into contact with fresh water will lack water channels, so the water moves so slowly that pressure doesn't become a problem. Other cells, like plants and bacteria, like I said before, have the cell walls that prevent the cells from expanding. This is why when people get IV fluids, they're dehydrated. You don't just get water. IV fluids have salt dissolved in them that's isotonic to our body fluids. We're going to talk about some examples of this later on in the year, but cells sometimes have to move materials against a concentration gradient. This is called active transport because it requires energy. Active transport of small molecules or ions across the cell membrane is done by what we call transport proteins or protein pumps that, again, are found in the cell membrane itself, in that fluid mosaic. Larger molecules can also be actively transported by these two processes, endocytosis and exocytosis. You don't need to go any farther than the prefixes to tell whether things are moving into or out of the cell. The energy that's required here goes into changing the shape of the cell membrane, either to swallow up something from the outside, bringing something in, as in endocytosis, or having a vesicle merge with the plasma membrane and release something in the case of exocytosis. These protein pumps are used specifically to move calcium, potassium, and sodium ions across cell membranes. And these changes in protein shape that happen, that require energy, play an important role in that protein pump process. This is a lot of the energy that goes into cells goes towards active transport. It enables cells to concentrate substances in a particular location, even when diffusion is wanting things to go the opposite direction. Endocytosis, like I mentioned before, is taking material into the cell by making pockets of the cell membrane. And then it forms a vesicle or a vacuole within the cytoplasm. Large molecules, clumps of food, whole cells can be taken in by endocytosis. There are two different types of endocytosis, phagocytosis and pinocytosis. Phagocytosis is large molecules like cells. Pinocytosis is smaller, large molecules like nutrients. A little bit more of a description of endocytosis here. There's pinocytosis. Large amounts of material released is exocytosis. And that's where vacuoles or vesicles fuse with the cell membrane and they release what's inside.